Guys, up to this point, we have had a discussion of replication of DNA. We're not really fully finished with it yet. And we said we've got to understand this in order to know how to replicate DNA from someone in a laboratory and how to analyze that DNA eventually on maybe a couple of pieces of equipment that we have at our disposal. So, with the replication, we said that we can start off with a DNA molecule, and this DNA molecule can be ripped up into two halves, and each strand is complementary of each other, meaning that as the new strand comes in, we are making a new copy, and a new strand will start to form, and those are basically made a new copy of what was here before. So we've taken a DNA molecule up here, and we've made a brand new one that's exactly the same. And then over here, we've made another one that's exactly the same. So what was one, we now have two. And then from here, we can rip those apart from each other. There we go. And here we go. And then we can bring in new strands to make new pieces. And this process will repeat itself over and over and over. And what was two is now four. And this continues on. This requires an extreme amount of energy, though. Not only that, but we have to rip these things apart. Well, do you remember the glue that I told you that we were worried about? This glue that holds the DNA strands together, those are hydrogen bonds. And we said there's tons of them. Think about it. 3.1 billion base pairs. 3.1 billion. And on average, they have 2 or 3 H bonds that connect those chains together. We're talking about tons of hydrogen bonds here. And those hydrogen bonds in that many numbers are going to be very complicated to rip apart. But this is why DNA is very stable. It's one of the reasons. We talked about a couple already, right? Hydrogen bonds leads to that as well. So we've got to come up with a way that we can separate the strands from each other, rip them apart, so that we could replicate them if we need to. And your body has to be able to do this too, right? So in this case, we can talk about an enzyme. And this enzyme is going to be called a helicase. And helicases need energy. And they get that energy from ATP. We've seen this before, haven't we? The A stands for the base pair adenine, the T stands for tri, and the P stands for phosphate. Remember on that slide, I told you, hey, look, we can have multiple phosphate groups, and as we rip them off one by one, we can get energy that gets liberated. Ding, 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 ding. Here you go. It's the same thing. So these helicases will rely off of ATP, adenosine triphosphate. And this is the energy source that it will need. And the helicases will go through and basically unzip the DNA strand together. All right, so here's the DNA strand, or at least one of the DNA strands. I'm just going to kind of double write it like this. And here we can start to unzip the DNA strand. So let me rewrite it. We go from here to here to here. And then maybe off of one, we get one strand that comes out. And maybe off of the other one, we get another strand that begins to unzip. And there's base pairs that hang off of here, right? There and there. And then in here are all of these base pairs that keep this molecule together. Helicases unzip the DNA. That's the easiest way to describe it here. Unzip the DNA. So, we now know the DNA strands will run in opposite directions. One of these will go from 5' to 3', but the other ones, the 3' is actually kind of down here because it runs anti-parallel with each other, right? So the direction of the strands are a little bit different. So as I unzip the DNA from the helicase, and I'm trying to copy the DNA strand, new DNA strands are going to be made in a particular way, and this is 5' to 3'. That means that one of the strands that we are talking about will be replicated, and this will be replicated under normal, well, I'm going to put in quotes, normal conditions. Nothing funny has to happen here. And this is called the leading strand. So as I unzip the molecule, one of those phosphate sugar backbones will be replicated in an order of 5' to 3' without a problem. Bases just start slapping onto that thing, one after another, after another, after another, after another. And that's what we call the leading strand, the one that's not going to give us any problems. However, the other strand will also be replicated because remember, we're taking one and we're making two from it. That will be replicated as well, but this strand kind of runs in reverse because of the anti-parallel nature of the sugar backbones. This one cannot be replicated continuously. This one is replicated in fragments. Small little segments at a time. It works on an area first, and then it kind of moves on, and it works on another area, and it moves on, and it works on another area as more and more get exposed. This is what we call the lagging strand. This one takes a little bit more effort. It takes a little bit more time in order to replicate. These small fragments, these have a name. And these are called Okazaki fragments. O-K-A-Z-A-K-I. Okazaki. Finally, this problematic strand here that has to be done in segments, the segments that are made have to be joined back together. It's got to be seamless. This is kind of the fine touch, fine tuning right here. And what joins these pieces together are something called a DNA ligase. A DNA ligase will glue it all back together for me. So one strand does not have a problem. It continues to replicate. It doesn't need anything special. It is in the proper order. It is very happy. Very easy to work with. The other strand, though, because it's the complementary version, runs backward. And because it runs backward, it cannot replicate in order. It has to kind of pick an area to start, replicate a small area, and then move on to the next. And it does this in terms of fragments that we call Okazaki fragments. And when all of the fragments kind of start assembling back together, they all need to be seamlessly connected. And that is the purpose of the DNA ligase that's down here at the very bottom. Now, what does the DNA ligase look like structure-wise? Did you really need to know? This is it. Folks, like we've said before, we are now talking about a different facet of chemistry, a different place in a laboratory. We are not talking about just a few carbons and a couple of oxygens anymore, like you've typically seen in general chemistry and even in organic chemistry. These things are huge. These things are so big that we don't even bother writing carbons and hydrogens or anything. We give you these computer simulation models of the 3D orientations and the shapes that these structures take on because there is so much. And this is the DNA ligase. What you're seeing here, the coils, there, and there, those coils are called alpha helixes. Those are not the DNA structure. That is the alpha helix that is within DNA ligase, meaning that when these atoms come together, they form these bonds, and these bonds begin to form a structure that will wrap around and around and around like a coil or a curly piece of hair. We also see these flat areas. There are these kind of purplishy ones that's here in the center, and those are called beta sheets. Those beta sheets are very flat areas of the molecule, or in this case, the enzyme, the DNA ligase. So we have these kind of plateau areas. When these atoms and these bonds come together, they're pretty much flat. And then we have these areas where, as the molecule begins to form together, it starts to coil upon itself in a 3D environment. But there's your ligase. Okay? That's the caliber of molecule that we're working with now. So these things are not very simple. These things are very difficult to look at, to monitor, to measure, and they're very temperamental. And what that means for me as an analyst in the laboratory is that if this is the stuff I'm working with now, there's a chance that it's probably not going to stay very long on a shelf, like an organic chemical. They're also very temperature dependent. So very often I'm going to have to put these things in a deep freezer or a regular freezer, or I'm going to have to keep them refrigerated. We are just working with a different class of compounds at this point. These compounds are very complicated. They're very complex versus something like benzene with just a ring and a couple of carbons, or the inorganic side of the house like sodium chloride. I can draw those all day long. I mean, I thought aspirin was bad to draw. Think about all the bonds that you got to go along with that. But now this? So again, very temperamental. The storage conditions are very important. And it's not uncommon for these things to have a very short shelf life before I have to reorder and reuse these materials in a laboratory. If I'm working in a forensics lab and I'm doing DNA work, very often I have to monitor the temperature of the refrigerators. I have to monitor the temperature of the freezers. I have to go through and I have to monitor them every single day and create a template or a spreadsheet of them. I have to be very particular with expiration dates. We have to get rid of things when we're supposed to get rid of things, because if these molecules, quote, die on me, they're not going to be able to do their job. And if they don't do their job, then I'm going to be wasting my time because of it. So here are these terms that we've just discussed, right? We talked about these Okazaki fragments. We talked about joining those together with a DNA ligase, and both of these are going to get replicated. But I never really did tell you how they were going to get replicated. The only thing that I said is that we slap on the bases, right? The molecule slaps the bases on that needs to go there, and away we go. Okay, well, the process is a little bit more complicated than that. Go figure, right? We knew it was coming. It's just a matter of how. So how does the strand, either strand, the lagging strand or the leading strand, get replicated and get back intact together like it was in the beginning? Okay, well, we need something in order to do that. And what we need is something called a DNA polymerase. The poly means many. So it is going to join many bases back together with each other. And these DNA polymerases are also very large molecules. Okay? They're actually a protein, very similar to a DNA ligase. And proteins are made up of many, 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 many amino acids. And amino acids are made up of many carbons and nitrogens and hydrogens and everything else in between. So this is another one of those structures. It's a 3D structure. It's a computerized model that we can kind of get an idea of the shape that these things take on. How many amino acids in total? We're looking at anywhere between 900 to 1,000 amino acids that are coming together to make this protein up that will do a specific job, and that specific job is to go to the DNA strand and begin to slap the bases on one after another after another. That's its whole purpose. But the DNA polymerase, due to the reaction and the way that it's made, can only add nucleotides, in other words, bases, to the 3' end of the newly forming strand. That's how we refer to that. And this will give rise to the 5' to 3' direction. So, in other words, it adds only to a 3' OH group. And it has to add in that direction. The problem is that no known polymerase at this point in time, no known polymerase can actually begin a chain. Something has to jumpstart it. Something has to be there to say, hey, polymerase, come on in and do your job. I'm glad you showed up to work today. So it needs a boss. It needs someone there to oversee this process that needs to go on. And this polymerase will need what we call a primer. The first two bases that we're going to refer to, the first two, are always RNA. But in order for this thing to do its job, it's got to have a primer that is there that will take care of that job to make sure that things get started. And then when that molecule shows up to work, then the other one can go away and leave it alone because it can trust that it's going to do its job for the rest of the day. So give me a bigger picture. Give me a better picture of all of this term and terminology. 5', 3', lagging strand, leading strand. What on earth are we doing? All right, well, let me see if I can help you out a little bit. So here's a picture of a DNA molecule that's getting ripped apart at this location, right there. Let me change the color so maybe you can see that a little bit better, right here. So here is the intact DNA molecule. You can see the base pairs on the inside here. The red dots represent the number of hydrogen bonds. We see one at the 5' end, right here. And if I follow that strand as it twists and as it turns, it looks like that's where it's going to end up. And this is the 3' end. Okay? On the other side of the molecule, I'm going to see the anti-parallel version. 3' is going to start here. And this band will wrap around itself, and I can track it. And away I go. There we go. And this is going to be the 5' end here. The sagey green color that you are seeing in the picture, this greenish color right here, is the new strand that's getting formed as the replication process is happening. And this is what we call the daughter strand. This was not part of the original DNA molecule before we ripped it apart. And because it's a new piece, it is kind of the offspring of that DNA chain, and we call it the daughter strand for that purpose. So one of the sugar backbones will begin to form a daughter strand, and the other one of the sugar backbones, if you see that here, that will also form its own daughter strand. So here is a depiction of a DNA molecule getting ripped apart. That ripping apart has to be taken care of by an enzyme called a helicase. And those things will begin to free up the strands on both sides of the DNA molecule. And then a DNA polymerase will come in, and the polymerase will start to replicate those strands as they are getting freed up. It will start to basically slap on the bases that you're seeing the daughter strand create. That's the purpose of the polymerase. Well, in order for that polymerase to do the job, we had to have a boss man that was there, and that was a primer. We had to have a primer for the ligase to come in and begin its job duty for the rest of the day. One of these strands had to be replicated in fragments. The other side of the house had to be replicated like normal. Not a big deal at all with them. Which side was which? Well, the side that runs in the opposite direction will need the Okazaki fragments. And those Okazaki fragments will be created in short little snippets. And then we get a DNA ligase that comes in, and it will glue all of those segments back together. Here's just another picture of it with some new terms. This fork that happens in the DNA is called the replication fork. That is where the strands begin to separate from each other so the replication can take place. We've also talked about this semi-conservative replication, and it allows basically for this chain to keep one half of the original in the first replication series at least, and it will always be continuous in the 5' to 3' direction. 5' to 3'. That's what you need to remember. The strand that goes from 5' to 3' is the one that the polymerase can sit on and basically replicate one after another base pairs. So in Blackboard, you're going to get a link. Here's the direct link in case you need to write it down or type it in manually. But you're going to see this link in the external videos. And if you go to YouTube and watch this video, it will put all of this into perspective in action. So again, it's going to be somewhat of a computerized model. It will kind of give you an idea of the process that happens here, and maybe you can walk away feeling a little bit better about the replication process that DNA has to take, so that way we can do our job in the laboratory and replicate a DNA sample from a suspect and analyze that DNA sample eventually on a piece of equipment that we'll have in the lab. So do not skip those external videos. I think they're pretty important. I think that it puts everything into context, and I think visually you'll see a little bit better the processes that have to happen in order for this replication step to go forward. And keep in mind, all of this is happening in our laboratory, right in front of our eyes, if we're adding the proper reagents and the other things that are necessary to get this done.